Hello and welcome to this video on the biomedical science pathway into medicine. So I'm currently a student that is about to go into the first year of biomedical science at the University of Auckland, and so essentially I'm just creating this video to share all the knowledge that I have learned researching and preparing myself for this year. So we're going to firstly look at just an overview of the course and then individually into each of the 4 core papers that are shared between health sciences and biomedical science, and then I'll also explore the 4 other non-core papers that are involved in the biomedical science course. Okay, so looking at an outline of what the year looks like. So the year is broken down into 2 semesters with a total of 8 papers. There are 4 core papers, and these are the same 4 core papers in the health science. And these are the ones in orange here. So Biosci 107, Chem 110, Population Health 111, and MedSci 142. So for biomedical science, there are 4 other papers, and health science will have 4 other papers as well. However, this video is only going to focus on the 4 other papers for the biomedical science course, as that is the degree that I'm personally going into. So in semester 1, we have Biosci 107, Chem 110, Population Health 111, and a general education course. And just to note, this is the Waipapa Taumata Rao course, and this is a compulsory general education course, and we'll go into what this involves later in the video if you stick around for it. During semester 2, the only core paper that we have is the MedSci 142. However, we also have Biosci 101, Biosci 106, and Physics 160. Okay, now looking at the entrance into medicine. So in order to be eligible for an interview, which is one of the core parts of being accepted into medicine, you need to achieve a minimum grade point average of 6.0. And so essentially the grade scale goes from a 0 to a 9, and an A plus being a 9. So a 6 is a GPA average of a B plus. And so you need a B plus average across all 8 of your first year courses with no fails in order to be eligible for consideration for an admission interview. However, not all those who have a GPA of 6.0 will get an interview. This is just the minimum requirement to even be eligible, full stop. So in order to actually receive an interview, this will be ranked off your 4 core papers. So that's the Biosci 107, Chem 110, MedSci 142, and Population Health 111. Another thing to note is that if your final grades are not available at the time of the interview ranking, an interim grade, so for instance for MedSci 142, you'll be given an A plus temporarily, and that will be used for the interview invitations. However, the official result will be used for the final selection. So in order to actually get us accepted and get an interview, you realistically need to aim for all A pluses. You can get in with just 1 A and 3 A pluses, but you shouldn't bank on losing any marks anywhere. You should try to go in with the mindset that you're going to get all A pluses. And just another important note is that you should submit your application to the Bachelor of Surgery and Bachelor of Medicine program during your first year of university study, no later than the 1st of July. So once you're in the course for biomedical science or health science, you need to apply then for your Bachelor of Surgery and Bachelor of Medicine during that first year, no later than July 1st. So that's quite important. And so the final rankings and whether you'll get accepted into this course will be 60% off made up of the GPA for the 4 core courses. The UCAT will count for 15%, and your multi-mini interview will count for 25%. Okay, so we're now going to look at the first, an overview of the first core course, which is Chem 110, and this looks at chemistry of the living world. Okay, so looking at the outline and the key topics for the Chem 110 course, so Chem 110, first off, comprises lectures, laboratories, and a range of self-paced online learning modules and activities. In terms of the key topics, we have the 5 key topics listed here with roughly what they're about. I'm not going to read through all of them. If you want to have a read, just pause the video. Okay, I've also listed here the 8 understandings for the Chem 110 course. They're a bit vague, not very that useful, so have a read of them if you wish. In terms of requirements, so the Chem 110 course folder is required for all students and can be purchased from the university bookstore, which is known as the UBIQ, and this contains the shell of lecture notes as well as the laboratory manual. I have actually looked at this bookstore and cannot seem to find these, so I'm not sure whether they come out just before the course or you get an email regarding how to get this. But you should find out before your course begins. Also, the Chem 110 lab manual is required for all students and can be purchased from the university bookstore as well. All students must have a hard copy of the lab manual, as you are not permitted to bring tablets or other computers into the laboratory. Okay, we're now going to look into labs. So labs is worth a certain percentage of your overall grade for Chem 110, which we'll get into the breakdown for what each thing is worth in a minute. However, just note that everything in the labs is actually marked and assessed and will go towards your final grade. So in terms of labs, you need to wear a laboratory coat, covered footwear, and suitable eye protection at all times. And students are expected to bring these to each session, and so you need to purchase your own lab coat and suitable eye protection and bring them to each lab session. And you can buy these from basically anywhere. However, they do have them at the university bookstore. Furthermore, in terms of the labs, they're broken down into 4 sections. So the first section is the pre-lab quizzes. And so these are very simple 10 multiple choice questions in which you get 10 attempts to complete them. And these are really easy to score well in. Just read your lab manual properly and knock it out in 1 to 2 attempts. The second part is the practical skills. So throughout the 10 lab topics, you're assessed on 10 different skills. So these are assessed by your teaching assistant, either when you present them your product or through assignment sheet you complete during the lab. The good thing about these is that you're assessed on every skill twice, which means you have 2 chances to get full marks. So keep a track of which skills you have done well on and which ones you need to try again with, and which experiments they correlate to. The third part is assignment sheets. So the first 3 assignment sheets are completed throughout the lab and are assessed, while the last 2 are not. So these often come down to the individual marker grading it, so make sure you get every detail you can on them and double check your answers so that the teaching assistants have nothing to pick on. Finally, you have a post-lab activity or the full report. So for the first 3 labs, you'll complete some written activities about aspects of your experiment. And then this builds up to a fill-in-the-blank scientific report in lab 4 and a full scientific report in lab 5. So make sure you complete the post-lab activities properly and take your teaching assistant's feedback in, because this will really help you for your final report. So another part of the course is the science and the media discussions, and so this will be a lecture about about the media and how they interpret science. And so essentially there are 2 things that you need to do to get a relatively easy 15% of your grade. So there are quizzes, and so these are similar to the pre-lab quizzes. There are a very simple 10-question multi-choice questions that you have 10 attempts at. The questions are derived from your SIM, which is Science and the Media, modules, but honestly, with some common sense, you can probably complete them without doing the modules. The second part is discussions, so there are 4 discussions. The first and last are worth 1 mark, and the other 2 are worth 2 marks each. In this, you will post a Canvas post to your discussion group about a certain topic/article that Caitlin outlines. Then you'll comment on another's post, and this is a super simple way to earn a free 15%, so make sure you do these on time. In terms of the quizzes, there are 11 quizzes throughout the course, and you only get one attempt to answer 6 questions on each quiz. This is anxiety inducing, but don't worry, they only take the scores from the best 8 out of 11 quizzes, so take your time on these and don't be afraid to consult trustworthy friends if they're willing to help. Okay, now looking at the test and exam, so there's a mid-semester test, and this test covers content from the first 4 modules as well as a few concepts from the lab, or labs, such as the yield calculations. There are 2 sections, with one being purely multi-choice questions and the other being short answer questions. The multi-choice questions are quite easy to do well in, but for the short answer questions, or SAQs, these are hard to do well in. And this is not because they are necessarily difficult, but it is quite hard to know what the examiner is looking for. Try to think about what the examiner really wants you to get out of the question, and also go on to the drop-in sessions to get feedback on your answers before the final exam. For the final exam, it is very similar to the mid-semester test in terms of structure. However, it covers all the content from everything, but there's very few questions about the labs. Do as many practice questions as you can, and make sure you nail your multi-choice questions if you need something to fall back on for your SAQs. You also will get cheat sheets for both the mid-semester test and the exam, and I've just got attached here an example cheat sheet from someone online. I don't know how good it is, but it potentially might be helpful for you in the future. Okay, so in terms of the scoring for Chem 110, so there are 5 different components, with the mid-semester test worth 15%, quiz worth 5%, laboratories worth 15%, assignment worth 15%, and the final exam worth 50%. So regular attendance at scheduled workshops will allow students to qualify for pulsage. So pulsage allows students to reweigh their test/exam contribution of their final grade from 1550 to 065 if their exam mark is higher than their midterm mark. So looking at this pulsage, this is a life-saving concept, which means if you do a certain amount of activities, you'll be able to relocate up to 15% of your test marks onto the final exam if you do badly on your test. So in terms of activities that you can do the first thing is the Best Choice website, and for this all you need to do is complete the activities on the Best Choice website. This website looks like it was created in the dinosaurs age, but trust me, it's really good. Use it to consolidate your knowledge and as practice questions, or just to brush up on hard concepts. In terms of the Best Choice, you only need to get 1200 points for the 15% pulsage, and the second thing that you can do is drop-in sessions. These are usually held over Zoom or in person in the lecturer's office, and this is a good way to ask any questions to the lecturers so that you know exactly what they're teaching. So just some more information, and so from here we've got lectures, and the teacher-led elements of workshops will be available as recordings. However, other learning activities, including labs, will not be available as recordings, so you must attend these. So just some study tips before we get into some general summary and overall tips. For both tests, start your cheat sheets early so that you can practice religiously with them. Get a feel for what sort of questions they ask through the practice questions they give you on Canvas. And in preparation for the labs, obviously you can't really practice making aspirin at home, but watching videos of people doing the experiments on Canvas helps to know the most efficient ways to succeed. Also, type all post-lab activities onto a Google Doc before submitting it in the Canvas text, and this way you can quickly refer to them for your final lab report and follow its structure. Okay, now just some general tips that I've found is that with the lectures, definitely buy the course guide, because it essentially just consists of all the lecture notes and lab notes that you'll need. Secondly, use Best Choice, so it's a good online resource for this. And the recommended textbooks do not seem to be that great, so don't bother purchasing them, and you can always access them online for free via Canva. In terms of labs, so how it works is that you'll have a pre-reading, a pre-lab quiz, you'll have an assessment sheet during experiments, which is handed in at the end of the lab, and then you'll have a post-lab quiz. So before going into a lab, look at the marking rubric. This essentially tells you how you're going to be marked, so you need to be able to tick all these off. The labs can be very time pressured, so you need to know what you're going to do before you go in. You need a lab coat, glasses, a $2 coin, lab guide, and stationery. And in terms of the tests and exams, they're multi-choice questions and short answer questions. And the best advice is just to do a lot of past papers and questions. Okay, so we're now going to move on to population health 111. Okay, so now looking at the outline and the key topics found in population health 111. So population health is just what it suggests, the health of populations. And the goal of studying population health is to improve the health of populations. This course introduces future health professionals to epidemiology, which is the science of measuring the distribution and determinants of disease frequency in populations and the effectiveness of interventions to control disease. Secondly, it is to the social and environmental determinants of disease and their distributions in populations. Thirdly, population health interventions to improve population health through health promotion and through the prevention and management of disease. And finally, number 4, the distribution, determinants, and control of important local and global population health. So, to be honest, that sounds like a whole bunch of nonsense, random words, and it's not really showing us what exactly we're doing in this course, so that's what I'll get into. Okay, so the understandings here will help us understand what we're actually doing in Population Health 111. So the first understanding says apply epidemiological measures to identify and describe the determinants of population health. The second understanding is to identify the determinants of health and apply this knowledge to describe the socio-demographic profiles of populations. The third understanding is to evaluate population-based and high-risk individual interventions designed to address population health issues and inequities. The fourth understanding is to critically evaluate particular approaches to improve population health locally and globally by using the course concepts to enable problem-solving and decision-making. The final understanding is to demonstrate academic integrity and develop team working skills and apply these to explore population health problems. So another component of the population health course is the PBL workshops. So PBL stands for problem-based learning, and it's just a fancy way of saying tutorials with a group project. Each tutorial also has a learning component with different topics to the lectures. In terms of the PBL workshops, the first sort of consists of age standardization, and this is probably the most math-heavy section of the entire course, with a few calculations you need to learn and memorize, and then apply in test situations. In terms of some advice, just spend a lot of time revising for this midterm test by going over each calculation thoroughly with practice questions, as this really helps consolidate your ability to memorize and use these calculations. The second is ethnicity coding. So this is a very simple concept where you just tally up the numbers of people who identify with a certain ethnicity and criteria, and then divide by the total number of people who identify in the same ethnicity group. And it's an easy way to get a lot of free marks in exams. The final part is the hidden matrix. So this is by far worth the most in exams, even more than lecture content. And in this, you'll learn how to identify different situations with different interventions in a table and be able to relate it to when it should occur in an event. So do a lot of practice with these and ask your tutors whether you're on the right track. So in terms of the PBL workshops, there's quite a few parts to these that are assessed with your group members. And so it's in your best interest to try and get those full marks, as it is quite easy to do so. So the first thing is meeting minutes. and the assessed activity. So each week leading up to your final PBL project, you'll have to meet with your group and complete the task on the meeting minutes sheet. With this, you'll each take turns to write minutes and lead the meetings, so you get your participation marks quite easily. In these sessions, you'll have to practice teaching your assigned multi-choice question for a class presentation, work on your PBL project, and then complete the assessed activity. Make sure you do the latter thoroughly and double check, as you can easily lose easy marks on this from not double checking. Second part is a multi-choice question teaching. So to gain your participation marks, you'll each take turns teaching a multi-choice question to the class. These are very, very easy, so don't worry, and it's a good opportunity to practice public speaking. These are also often similar to questions you may get in mid-semester test and exam, so take it as an opportunity to revise as well. In terms of the PBL project, this can either go really well or really terrible depending on your group members. However, it's quite easy to gain full marks as long as you follow the rubric. Once your presentation is done, so you've gave your presentation, you'll be asked a question by another group, and you'll be assessed on how well your group handles that question. They're quite lenient with this, so don't stress out too much on what people could ask. Everyone gets the same mark for this, irrespective of the amount of work they do, unfortunately, but use your group evaluation to roast those who aren't doing their share. Finally, in terms of participation marks and group evaluation, this is the easiest 5% you'll ever get, so make sure you do your fair share of work. You'll also get to evaluate your peers so that everyone gets the marks they deserve. In terms of the quizzes, there are 6 multi-choice quizzes, each with 5 questions that contribute 6% to your final grade. You have 3 attempts at each with a 10-minute time limit. The bad thing about these quizzes is that once you submit an attempt, you won't be able to see the questions or answers to them until, you won't be able to see the questions or answers to them again, and you won't know what you got for each attempt until the grades are released at the end of the week. Luckily, however, these are quite simple and match quite well to the actual multi-choice questions you'll get during the mid-semester exam. So now looking at the test and exam. So firstly, for the mid-semester test, this is a 40-question multi-choice question test where each question is worth 1 mark. This test is normally a week after the BioSci 107 test, so you won't usually have much time to study for it. The content changes year to year. For example, in 2021, they had questions on the first module, half of the second module, and 4 questions on age standardization. But this will change year to year depending on how fast the content gets through. The only multi-choice questions that can kind of trick you is ROD section, so make sure you read those carefully. Otherwise, this is a fairly straightforward test with nothing too surprising if you can internalize the concepts from each lecture. Looking at the final exam, this is purely made up of short answer questions with marks for each question ranging from 0.5 to 5. The questions aren't necessarily difficult, but you need a good, well-rounded understanding of all the lectures. The big money questions worth a lot of marks can vary year to year, but one is usually the Haddon matrix. In terms of some advice, luckily in population health, everything you need is on the slides, so you don't have to listen in too closely to what the lecturers are saying. And almost all of the pre-readings, except for the ones that they tell you will be in the exam, are basically useless, so don't read them unless you want to know more about the case studies Rod's talking about. So in terms of the scoring, there'll be a midterm test worth 20%, workshops worth 25%, an online quiz worth 15%, and an end-of-course assessment worth 40%. So in terms of some more information, this course does not have a prescribed textbook. However, they advise students to read the Essential Epidemiology, 4th edition, which is all outlined there. And lectures will be available as recordings, but other learning activities, including workshop and office hours, will not be available as recording. So just some general tips for Population Health 111. In terms of the lectures, do purchase that textbook that is recommended. It is great. Do all the pre-reading as you are tested on them. So throughout the course, you'll be given pre-reading stuff. You should do these as you will be tested on them. And there's also a PDF called Gate Notes, and you should learn these Gate Notes really well, as you must understand these concepts really well, as they'll try to mix up the wording and sort of things like that. So if you have a really good understanding of these concepts, you'll be fine. In terms of the tutorials, you have tutorials where you will reinforce and add some information from lectures. You also will do a research project with about 5 people. And in terms of this research project, really do your share, as your peers will be grading you on how much work you've done and the quality of your work. In terms of the tests and exams, the midterm exam is only multi-choice question, and this is where you need to watch out for the subtle change in wording, which is why those gate notes are so important to be really well versed on. In terms of the exam, this is mainly short answer questions and fill in the blanks. And that's really it for Population Health 111. Moving on to biology for biomedical science, and we're looking at cellular processes. This is BioSci 107. Okay, so as an overview to this course provides an introduction to human biology with a particular emphasis on the cellular basis of form and function, cellular components and processes of blood, neural, muscular, immune, and supporting systems will be discussed. So the key topics, the first topic is cells and tissues, the second is cell structure and function, the third is special topics, the fourth is blood and immune, the fifth is cell processes, the sixth is excitable tissues, which are neurons, and the seventh is excitable tissues, which is muscles. In terms of understandings, the first understanding is to identify and describe the structure and function of human cells and tissues. The second understanding is to define and describe the cellular processes that enable human cells to perform required functions. The third understanding is to use and apply practical laboratory equipment correctly to collect experimental data. The fourth understanding is to critically evaluate experimental data, presenting findings clearly and logically. The fifth understanding is to participate in group discussions, communicating effectively and logically. Okay, so looking at the labs for BioSci 107, there are 5 labs split into 2 parts each. So the first part is a 5-question online multi-choice question quiz, and that you will complete this before the lab. And then the actual practical lab where you fill in a sheet with short questions. So in terms of the 5 labs that there are, the first one is histology, the second one is embryology, the third one is hematology, the fourth one is osmolarity, and the fifth and final is muscle function and fatigue. Okay, so now looking at online feedback activities, so there are a number of activities that can be done to earn a free 10% towards the course. So this is a great way to engage with learning as well as help boosting your overall grade for BioSci 107. There are 12 different activities that can be done, and the best 10 contribute towards your final grade. So the first way is use Wiley Orion Plus. So this is a really old website, but it complements the Tortora textbook very well. You just need to do a couple of questions on one objective to be able to gain the 1% towards your extra 10%. So also use this for the first module, as some of the questions from here turn up in the mid-semester test. The next way you can do it is by writing and commenting on one question on pairwise relevant to firstly the first 3 modules and then a second percent for the remaining modules. So this website is also quite old, but it's a pretty simple task. First, you need to write a question relevant to the first 3 modules, and then for the second one to the rest of the modules. And then you need to comment on another person's question by just giving them positive feedback. And this is another way to earn a simple 1% per 1% for one for the first 3 modules and 1 for the remaining modules, like I've said. And some people also use this resource to consolidate their learning for exams. In terms of the quizzes, so there are 7 quizzes, and for these quizzes, you'll answer 10 multi-choice questions. Each question varies in difficulty, with the first few being very simple and the last ones being more like possible exam questions that you might have. You get 3 attempts, so use the first one to test your own knowledge without any help, go back and review which ones you got wrong, and then attempt it again with your notes available. In terms of the mock test and mock exam, so these are both held on Canvas and are similar to the test you'll get when you sit it after the semester break. And you have unlimited attempts on it, so use the first attempt to get an actual trial run and make sure you fill in the gaps as the break goes along. Okay, so in terms of tests and exams for Biosci 107, the mid-semester test is probably the first university exam you'll ever sit in your lifetime, and you should buckle up because it is a roller coaster. In this test, you have 70 multi-choice questions in 90 minutes. The test only covers the first 3 modules and the first 2 labs. You'll also be able to bring in your cheat sheet to help you out through the exam. The lab content usually only tests on theory, but sometimes you have to remember equations from the first lab, so you should put these on the cheat sheet as it's so easy to do so. Also, be aware that the first module's questions are often poorly worded, and these leave many people stressed out and scratching their heads after the exam. Definitely read the questions really carefully. For the final exam, it's similar to the mid-semester test where the questions are purely made up of multi-choice questions. You have 2 hours to answer 80 questions which are based on only the last 4 modules and last 3 labs, so there's nothing from the first test. Make a good cheat sheet and go over lots of past exam papers as they tend to reuse similar questions each year. In terms of the scoring for Biosci 107, the laboratories make up 20%, quizzes make up 10%, the test makes up 30%, and the final exam makes up 40%. Just some more information is that lecture attendance is strongly recommended. However, they're available as recordings, but other learning activities, including labs, are not available as recordings. The course guide is available to purchase through the University Library UBIQ, or it can also be downloaded for free via Canvas. But if you are downloading it, you need to bring the relevant pages from the laboratory manual to each of your practical sessions. You have to print them out. The prescribed text is listed there. This is also the prescribed text for MedSci 142. And an additional supporting textbook is also outlined there. So just some general tips. In terms of the lectures, the course guide is not that useful, so don't rely on it too much. Second point to really note out is that this is a really content-heavy paper. And another important tip is to, before you look at the minute details, zoom out and understand the process or the system as a whole, and then zoom in and understand the little minute details. And this will help you understand it better. Next, the textbook is very helpful, so use those textbooks. In terms of labs, it is a very similar format to Chem 110, but it is a much more relaxed environment. And in terms of the tests and exams, both are multi-choice questions. So we're now going to look at the final core course for biomedical science, and that is biology for biomedical science organ systems, which is MedSci 142. Looking at an overview of MedSci 142, so this is an introduction to human biology with a particular emphasis on integrated organ function. So this course will deal with structures and processes associated with the function of the nervous, locomotor, cardiovascular, respiratory, digestive, renal, endocrine, musculoskeletal, and reproductive systems. In terms of the understandings, the first one is to explain in depth the importance of particular organ systems. The second is to explain how any imbalance can affect health and lead to disease. The third is to use and develop your intellectual, cognitive, and practical skills to complete the learning and assessment activities. The fourth is to communicate your knowledge and understanding as a future healthcare and/or scientific professional with fellow students, the academic faculty, and the community. And the final understanding is to plan and evaluate your own progress towards achieving personal and professional goals. Looking at labs for MedSci 142, so instead of getting marked on your experiment or filling in a sheet like Biosci 107, you have a 10-question multi-choice test to set at the end of your lab session, and your best 5 out of 6 scores count towards your graded 10%. So the first lab is on digestive system and body cavities, so you'll do a rat dissection. The second is on the human brain. The third is on the mammalian heart, so you'll do a sheep heart dissection. The fourth is on the reproductive systems, the fifth is on the ventilation laboratory, and the sixth is musculoskeletal system, and you'll do a chicken leg dissection. Okay, now looking at the online activities and quizzes. So this is probably the easiest 20% you're ever going to earn in this course. So make sure you do all of the activities to get full marks. Not doing them is basically throwing away any chance you have of reaching the next grade boundary, which could prove crucial. There is one activity on Coral Cloud after every lecture that are due the week afterwards. Try to do these properly without your notes, as you won't be able to do that if you get in-person assessments. The 3 with the lowest marks will not get counted towards your final 20%. The online quizzes are 10 multi-choice questions due at the end of each week. You fortunately get unlimited attempts to make sure you get those full marks. All the questions from the multi-choice questions are taken from the PDF on Canvas, so do those first, confirm your answers, then attempt the quizzes online. Now looking at the tests and exams for MedSci 142, there are 2 tests and 1 final exam. The first test is purely multi-choice questions with 52 questions on the nervous, autonomic, endocrine, cardiovascular and reproductive system lectures, as well as content from labs 1 to 3. The test is 90 minutes long, and the practice questions that are given are mostly straightforward and should reflect what you get in the test. In terms of test 2, this has the exact same structure as the first test, except it was on the respiratory, renal, musculoskeletal, and digestive systems with content from labs 4 to 6, and it's also 90 minutes long. In terms of the final exam, it is purely short answer questions worth 228 marks, and this tests you on all the lectures with a very short section on integrated knowledge from lectures and labs, and this in-person exam is 3 hours long. Looking at the scoring for MedSci 142, the labs are worth 20%, the online activities are worth 10%, the tests are worth 40%, the final exam is worth 30%, and that makes up the overall 100%. Just looking at some more information, you should always use your lecture notes as a guide on what to read in the textbook and what to focus on for assessments. MedSci 142 is necessarily a content-heavy course which aims to prepare students for the rigor of subsequent studies, including professional and clinical programs. Technology permitting, lecture recordings will be available. Other learning activities, including labs, will not be available as recordings. Test 1 is always held on Monday of week 7, so this is the 15th of September. And test 2 is always held on Friday of week 12, which is the 24th of October. Finally, the recommended textbook is Tortora and Derrickson, which is the Principles of Anatomy and Physiology. Alright, so I'm now going to move on to look at the non-core courses in the biomedical pathway. So if you're doing the health science courses, these will not be a part of your course. And just to remember, you need a B+ average in all of your papers to be allowed an interview. So even if you have all A+ in the core papers, you have to do still decent in these non-core papers to be eligible for an interview. Okay, so the first non-core course we're going to look at is the general education course. So this is Waipapa Taumatu Rao, which is Contemporary Science in Aotearoa, New Zealand. And this is WTR Sci 100. So looking at an overview of this course, it considers how knowledge of place enhances your learning. It looks at the significance of Te Reo Waitangi and how knowledge systems frame understanding. So in this course, we cover a wide range of topics, including AI and ethics, astrophysics and place, philosophy and the meaning of science, professional ethics and science communication, fast fashion and sustainability, and science in context, including Te Reo Waitangi. Okay, now looking at understandings. So the first understanding is to demonstrate how place and an understanding of Te Reo Waitangi are significant to your field of study. The second is to critically and constructively engage with knowledge systems, practices, and positionality. The third is to employ a reciprocal, values-based approach to collaborating. The fourth is to communicate ideas clearly, effectively, and respectfully. The fifth is to reflectively engage with the question of ethics and academic practice. And the sixth is to demonstrate a critical understanding of sustainability. So looking at the key topics for this course,
In week 1, we look at whangawanatanga. In week 2, we look at te tiri o waitangi. In week 3, we look at knowledge of place. Week 4, we look at what are knowledge systems. In week 5, we look at knowledge systems in science. In week 6, we're working across knowledge systems in science. In week 7, working across knowledge systems beyond science. Week 8, developing an ethics toolkit for science. Week 9, ethics in practice. Week 10, communicating knowledge. Week 11, te tiri o waitangi in science and contemporary Aotearoa New Zealand. And week 12, ka moa ka muri, so this is reflecting back. Okay, so some more information is that there's no textbook for this course. All learning resources are free and available online through Canvas once the course begins. Each week, students are expected to complete an online module comprising of short videos, readings, and activities, and students can expect to spend about 2 hours completing the online module each week. The studios are 1 hour per week and labs are 1 hour per week, in person only, and attendance is required. Active participation in studios and labs is a graded component of this course, and studios and labs are not recorded. In terms of the scoring, the studio participation and preparation is 20%, the online module activities is 20%, the effective teamwork is 10%, reflective activities, which there are 2 of, is worth 15%, assignments are worth 25%, and presentations worth 10%, and this makes up the overall 100%. Okay, so we now move on to our next non-core course, and this is Life, Origins and Mechanisms, and this is Biosci 101. Looking at an overview of Biosci 101, this course questions what life is and explores its machinery. It speculates on how life arose from the flow and capture of solar energy to power growth, movement, replication, and storage of genetic information. It describes how genes interact with the environment and how mutations can be catastrophic or transformational. These processes underpin life as we know it. All teaching activities and resources can be accessed in Canvas. The lectures guide you to learn knowledge and the process of answering assessment questions. Practice activities with feedback and questions are available online in the tutor system Mastering Biology. The Mastering Biology system gives you access to the e-text version of Campbell's Biology, which is a prescribed textbook. The knowledge you have gained during the course is assessed in a short test in week 10 and a final exam. Laboratories occur 6 times during the semester, and these are fortnightly and are also assessed. Looking at the understandings, so the first is to identify, describe, and explain the key concepts and processes of biogenetics, cell biology, and genetics to the level required for progression to Stage 2 biological science courses. The second understanding is to demonstrate proficiency in constructing and interpreting graphical and numerical data. The third is to demonstrate core competency in basic biological laboratory techniques. The fourth is to use active learning approaches to become an independent and passionate lifelong learner. The fifth is to follow self-directed processes to monitor and regulate your learning to develop academic skills and intellectual independence. Finally, number 6 is to work responsibly, safely, and ethically, both individually and in peer or team contexts. Okay, so looking in more depth at the key topics. So the first is bioenergetics. So the lecture topics include an introduction to biochemistry, thermodynamics, and its relevance to metabolism and life, the chemical components of cells, glycolysis, the citric acid cycle, the second of Krebs cycles, the electron transport system, the spark of life, ATP synthesis, the search for the squiggle, glucose, eat it or make it, blood glucose levels during the fed and fasting states, exercise and diabetes, photosynthesis, the conversion of light energy to chemical energy, or knowing where French fries really come from. Second, we're looking at cell and molecular biology. So this topic includes cells and organelles, cell membranes, cell junctions, and cell membrane transport, the structure and composition of proteins, enzymes, and the nature of enzymatic catalysis, structure and function of nucleic acids, synthesis of mRNAs, that's transcription, and protein synthesis, that's translation, the genetic code and transcriptional control, the control of gene expression in bacteria, so we're looking at the lac operon.
Finally, we look at genetics. So this topic includes the genetic basis of inheritance, population genetics, how genes interact with the environment, DNA technology, including the latest DNA sequencing techniques and their ethical implications, biotechnology, and gene editing, personalized genomics with a focus on research applications in New Zealand. Looking at more information, so students are required to supply and wear a lab coat and safety glasses while in the teaching lab. Lectures will be taught on campus and available as recordings. However, other activities, including labs, will not be available as recordings. The prescribed textbook is the Campbell Biology, as seen there, and other recommended reading is available through the library course pages. And the course workbook and laboratory manual can be purchased from UBIQ, that is the University Bookstore, and this book is also available in Canvas and electronic format. Finally, just looking at scoring, laboratories are worth 28%, the online formative assessment is worth 12%, the test is worth 20%, and the final exam is worth 40%. Alright, so we're now looking at Biosci 106, which is the foundations of biochemistry. So looking at an overview of Biosci 106, so this course gives an introduction to the core elements of biochemistry, investigating biological processes at the chemical and molecular level. Key themes within this course include the molecular structure of proteins, enzyme kinetics, biochemical energetics, carbohydrate and lipid metabolism, nutrition, cell signaling, vision, and aspects of plant biochemistry, including world food production. These themes provide a framework for discussion of mechanisms underpinning human disease, including diabetes and obesity, antibiotic resistance, drug development, and plant medicinals. Looking at the understandings now, so the first understanding is to identify, explain, and evaluate selected, fundamental, universal concepts that drive molecular interactions and processes in biological systems. The second understanding is to identify and describe core chemistry concepts, the role of biological catalysts and bioenergetics that are fundamental for synthesis of biomolecules and processes in living systems. The third understanding is to define and describe the synthesis and role of lipids and adipose tissue in biological systems and disease. Number 4 is to explain and evaluate the role of nutrients in common biochemical pathways and disease, the relationship between bacteria, their host, and development of antibiotic resistance. The fifth understanding is to identify and describe how cells recognize and respond to changes in their environment, as well as the mechanisms of signal transduction and the role of signal transduction in disease and drug design. Number 6 is to use and apply scientific practical skills in a safe and effective manner and be able to critically evaluate, interpret, and communicate experimental results. And finally, the last understanding is to develop and demonstrate the ability to work skillfully in a collaborative setting, communicating effectively to present scientific information in an articulate and rationale way. Okay, so looking at the key topics, so Module 1 is Chemistry of Life. So the basic concepts of chemistry and universal rules that define the biology of life. Module 2 is Catalyzing Life, so the concepts underpinning biological catalysts that drive biological processes. Module 3 is Energetics of Life, so the bioenergetic principles linking life's origins with all contemporary biological life forms. Module 4 is Lipids, Metabolism, and Obesity, which is the role of water-insoluble molecules in biological processes and in medical conditions. Module 5 is Micronutrients and Antibiotics, so the ubiquitous role of vitamins in biology and the interplay between host, pathogen, and medical interventions. And finally, Module 6 is Signal Transduction, and this is the key concepts underpinning how molecules and cells talk to each other. Just some more general information. Students are required to supply and wear a lab coat and safety glasses while in the teaching lab. The Biosci 106 course book, including a lecture guide and laboratory guide, can be purchased from the UBIQ, which is the University Bookstore. Both are available in Canvas and electronic format, and you're required to bring the relevant pages to each laboratory section. The recommended textbook is Appling et al. 2019, Biochemistry, Concepts and Connections, Global Edition, ebook 2E. Looking at the scoring for this course, so quizzes make up 12%, laboratories 28%, the test 20%, the final exam 40%.
Okay, so now I've just got some general tips for both Biosci 101 and 106. So in terms of lectures, the course guides are very summary-like and are not really worth buying, so just use the online Canvas version. However, the textbooks are very useful. In terms of labs, timing is key. There is a lot of time pressure, so make sure you plan out what you're doing really well. And also listen to all the tutors as well, as they will give answers. And even if, for instance, they're in the middle of a lecture and they start talking, try to focus on what they're saying more so than what you're doing, or sort of pause what you're doing a little bit, because they'll often give out answers that are really crucial. And finally, in terms of tests and exams, they're both multi-choice questions. However, they're not just fact recall, but more understanding based. And the content for the mid-semester test is not reassessed in the final exam. Okay, for the final course, Physics 160, this is Physics for the Life Science. An overview of this course. So this course covers 5 subject areas. That is mechanics, thermal physics, fluids, optics, and waves, electricity, and medical physics. The medical physics topics, which are lectured by staff from the School of Medicine, include biomechanics, fluid mechanics, audiology, vision correction, and medical imaging. And the course is primarily algebra-based and includes lectures, laboratories, and tutorials. In terms of the understandings, the first understanding is to explain the physics behind many biological systems using physical concepts from mechanics, optics, and waves, thermal physics, and electricity. The second understanding is to apply specific skills and methods used in physics to solve problems. The third understanding is to apply the laws of physics taught in this course to algebra-based problems. And the fourth and final understanding is to contribute to group-based problem solving and experimental work. Just some more information is that lectures will be available as recordings. However, other learning activities, including labs, will not be available as recordings. In terms of the course book, this is provided by the course, and the textbook is Physics by Cutnell and Johnson. In terms of the scoring, assignments worth 10%, test 1 is 10%, test 2 is 10%, laboratories are 20%, and the final exam is 50%. Some general tips. So the textbook is very helpful for the lectures. In terms of the labs, the practical investigation points, so you get points for format. So this is titles and being cleared to read and things like this. And then also things in the marking rubric will get you points. Instructors will tell you what to have or what you have to have in the report. So listen to the instructors during the lab. In terms of tests and exams, both are multi-choice questions. Just do many practice questions to get familiar with all the calculations and concepts, and the exam covers all the content. Okay, just some final notes. Thank you all for watching this video, and I hope you found it helpful. Bear in mind that this is information I was able to find, and every detail might not be exactly correct. However, it should give you a general idea of what is involved in the course, what is involved in each of the core and non-core courses, and hopefully it will give you a helping little head start, and there are some tips you can come back to when you start each course. Furthermore, the link that is on this page right now, if you type that into online, you'll be able to find some more decent advice or a shorter overview of the core concepts, and I hope you find those helpful as well. So that's pretty much all there is. In terms of the other parts of getting into medicine, which is the MMI and the UCAT, I'll have another video coming out soon about the UCAT 2025 and onwards with the new adjusted changes, and also later in the year I will make one about the multi-mini interview. But that's this for now. Thank you for watching the ultimate guide for University of Auckland biomedical science.